Good morning, ladies and gentlemen. Welcome to Quant-inar, organized by Elucidator. Today, you will learn about the quantification workflow on poly and how to quantify large number of metabolites by going from peaks to absolute concentration in minutes. Let's quickly look at today's logistics before we move on to the presentation and the demo. This webinar is being recorded and a copy of this recording will be shared with you over email. We will take all the questions in the last 10 minutes of the webinar. You can post any questions you want us to answer in the question answer session at the end. We also have a support team here, so if you have any doubts or questions during the demo, you can post them on the chat or raise your hand and they will reply promptly. So let us start with the introductions. For today's webinar, we have with us Dr. Sean M. Davidson presenting how he has used Confit in his study on understanding the pharmacokinetics of cysteamine in rats. To tell you a little about Sean, he did his Bachelor's of Science in Biology with a minor in Mathematics from Providence College, followed by his PhD in Biology from MIT, where he worked at the Vander Heiden Lab in the domain of oncometabolism. He then did his postdoc from Broad Institute, followed by which he became a Louis Sigler Independent Fellow at Princeton University and now a Principal Investigator at Princeton University. Throughout his academic career, Sean has worked on some very interesting problems in biology, which is evident from the over 20 publications he has in journals like Nature, Cell Metabolism, amongst others. In today's demo, Sean will take us through one of his studies which he has performed using the quantification workflow on poly. As for me, I'm Raghav Segal, product manager at Elucidator, and I work with both Almaven and Poly. I've been author to a few publications in Nature and Nature Methods, mostly centered around using Almaven and Poly and how they can be used to understand metabolism of cells better. I've also been author in multiple posters at ASMS, CPSA, and TCGA. Today, I will be talking to you briefly about Elucidator, Poly, and Almaven, as well as give a short demo on how to quantify large number of metabolites swiftly before handing over the webinar to Sean for presenting his study. We also have the Poly support team that consists of Surbhi and Nikita, Associate Product Manager, Shubham, Data Engineer for Poly, Shubhra, Technical Lead Almaven, Heman and Omkar, who have been actively onboarding users to Almaven and Poly. They will be answering all your questions throughout the webinar. Without further much ado, let's briefly talk about Elucidator, Poly, and the Quantifit workflow. So to start with, who are we? We are Elucidator, a 50-member strong team which is based out of Cambridge and New Delhi with over 25 partners. Our expertise in target discovery, metabolism, and big data tools. Cumulatively, we have over 5 publications in high-impact journals such as Nature, Cell Metabolism, amongst others, as well as over 10 presentations in conferences such as ASMS, TCGA, CPSA, amongst others. All of this we have achieved in our limited period of existence of just over 3 years. Our goal as a company is to bring cell metabolism capabilities to both industrial and academic labs. As a company, we have multiple arms. One of them being services where our bioinformaticians, data scientists, and biochemists help labs in discovering new metabolic insights using cutting-edge tools and data. We have tried to build these services and processes into our products, which will allow scientists at labs to directly discover insights without our help. Poly was essentially built on this idea. We imagined it to be a platform for a metabolism lab. Let's watch a brief video about Poly to understand more. saw in the video, Poly tries to address different barriers to omics analysis, whether it being by easing process of varied large omics data or making experiments cheaper by using publicly available data. Poly offers customized and robust workflows which are accessible anywhere at any time and enable you to get to pathway level insights with ease. As a platform, Poly solves challenges of communication and collaboration as well. All of this makes Poly the ideal platform for the metabolism lab. In its beta mode, spanning approximately an year, Poly has gained a lot of traction. It grew 50x in its user base, while at the same time growing 200x in the number of datasets processed per week. All of this came from users in over 60 labs in both industry and academia. By the end of this year, we plan to have 7 workflows on Poly, ranging from mass spec omics to sequencing omics to integrated omics. These workflows can be used for both hypothesis generation and validation. In the forthcoming weeks, we will be giving webinars on a few other workflows on Poly, so stay tuned in. Now let's talk about the Poly Config workflow. Poly quantification workflow goes from raw metabolomic data such as MZXMLs up to concentration plots of your quantified and normalized metabolites. And not to forget, all of this can be done in minutes. And that's not it. The quantification workflow can work with any kind of mass spec method, whether it be labeled or unlabeled, MS or MSMS, LC or GC. Quantifit can handle all of them. Now, let's get down to actually talking about how to quantify large number of metabolites swiftly using poly-config workflow. Now, before we actually get into the demo, let's briefly talk about the data we will process today. The data that we have is high-resolution LC-MS data. It has approximately 25 metabolites with standard samples. A total of 43 samples amounting to 2.52 GB in size are present in this data. The 43 samples are of 4 kinds essentially. 9 pooled standard samples, 3 blank samples containing just the buffer, 6 QC samples which are pooled from unknown samples, and 25 unknown samples. Now, the first step of the quantification workflow is peak curation, and for this purpose, we are using LMaven, a mass spec data processing engine, with its ability to do automated peak curation, process large data sets, and handle data from all types of mass spec machine, while being vendor neutral and displaying ease to interpret visualization, makes it a one-of-a-kind tool which is enabling the quantification workflow on Poly. LMaven has grown significantly over the years, and as you can see from the 700 downloads we have acquired in a span of just 8 months. LMaven has also been accepted for a book chapter for methods in molecular biology. Also, we had recently done a webinar on LMaven, the link to which is being provided to you in the chat. Now let's actually talk about demoing the demo where I show you processing of the quantification data through LMaven and ion count normalization on Poly. Now here I've moved to my machine. For doing this, I will open LMaven, of course, first, where I will upload my sample. Now I can go to open here and upload my samples. As I said, there are 43 samples here. Uploading all my samples will typically take a few seconds. And as I just mentioned, there are 43 samples which amount to 2.5 GB in size. As you can see, LMaven uploads this data very quickly. An important point to note here is that the compound data of my choice has already been loaded. Of course, to know more about how to use LMaven, refer to the LMavenar link provided to you, or go to our YouTube page to view tutorials on LMaven. So now that the data has been uploaded, I will directly perform peak curation in LMaven, for which I will go to the peaks tab and click on find peaks to automatically detect peaks in my data. This will take a few seconds. Now that the peaks are here, I can scroll through them. But since I know LMaven automatically curates peaks with very high accuracy, I'm going to move to my downstream processing. To know more about accuracy measurements, refer to the poster we presented at ASMS, which has been added in your handouts. Now, moving on to the downstream processing, we will now take this data to Poly. we can possibly take this data through the LMaven Poly integration, which is what I'm showing right now. But for this, we will have to first go to first view, a gateway app into Poly, and then into Quantfit from there. Since this might take some time, we are going to manually upload data from LMaven to Poly. Having said that, the next few releases of LMaven will see the direct connection between LMaven and Quantfit also being built. Now, to manually export the data to Poly, we will first export all the groups in the CSV format, which I'm highlighting right now. And I will select all groups to be exported since I want to export all of them. I can save it by whatever name I want in my downstream data processing. I've already done that in my lmaven output.csv, so I'm going to skip this step. Okay. Now, I will now go on to my Chrome browser and log into Poly. This is, of course, the pre-login page of Poly. I have already put in my credentials and I'm going to submit them. Once logged in, we would land on the Poly dashboard, which shows the whole palette of apps a user can choose from. We're going to use Quantfit app, which is available here. Once we click on the app, we are prompted to fill a project name, which I'm going to use. In our case, I'm going to add a new project called Poly Quant-inar. Oh, yeah, Poly Quant-inar. And I'm going to run make my project and run. This will land me to the Poly Quantfit upload page. Once on this app, I can go to the documentation if I like, which is on the top right hand corner, or download the demo files from here. But since I already know that I need to upload two files, I'm not going to open any of this. The first file I need to upload is the LMaven output file, of course. And the second file I need to upload is the standard metadata file, which will map the standard samples to their concentrations. I'm going to show this file just in a minute while the data is being uploaded. So as I was telling you about the standard metadata file, it has 5 columns in it. 2 of them essentially refer to the standard name. We also have the concentration column for every standard. So every standard is mapped to a particular concentration along with its unit. And for every metabolite, this particular standard and its concentration has to be mentioned. One can also sort of change the concentrations for different metabolites, something that our users often do. Now, once the data has been uploaded, and this takes a couple of seconds, I just wanted to show you the metadata file. And as soon as the data gets uploaded, we reach the injection efficiency correction normalization. Here you will, this whole ion count normalization or injection efficiency correction normalization, as we call it, will enable you to normalize your data with respect to internal standards and correct for any injection inefficiency errors. To use this normalization, you would have to add an internal standard in same quantities across all your samples. This is something that you would have to do while preparing your samples. And you will then come to Poly and select the internal standard, which can be anything which you have chosen. Poly will automatically look at the intensities of all the samples for this particular standard and determine outliers which should not be used for calculating factors. Once that is done, Poly will give you the option to select a particular sample to calculate factors by dividing the intensity of this sample with all the other samples. These factors can thus calculated will then be used to normalize all other metabolites. For this particular analysis, we are going to skip this step. The processing time for this is typically 30 seconds from normalization to the dashboard. In the meanwhile, I will show you a bit about the features of Poly Quantfit. A lot of them I've already been talking about, such as its swiftness and the ability to go from raw data to custom plots. But there are a few other key features that Quantfit offers, such as the ability to perform normalization, and not just ion count normalization, which is prior to quantification, but also the ability to normalize your data post-quantification with respect to a metabolite, or even scale the data by a certain factor. Quantfit Workflow gives you the ability to quantify your metabolites using 5 different curve fits, essentially linear, polynomial, exponential, log, and par. Users can also further tweak these in specific parameters for each fit. Quantfit allows you to reject outliers on the fly. It is, of course, linked to IPython scripts and a variety of other apps on Poly, which will allow you to customize the workflow as you like. Quantifit allows you to save and restore your analysis to the exact state you left your analysis at. No need to worry about that reproducibility anymore. Last of all, users can share their quantification analysis with their collaborators to discuss and deliberate on their analysis and data. We are, of course, going to show you all of these features and more on Quantifit now. Now, as you can see, we have landed on the linear fit data. As soon as you land on the Quantifit dashboard, you can see that all of the data has been linearly fit. And we can see which metabolites have been fit well and which of them have not been fit well by looking at the metabolites by looking at the metabolites using the downward arrow at the metabolite carousel. Anything with marked with a red is a bad fit, and you on hover you can see the R square of this particular metabolite. You can also fit these metabolites better, as I can see that there is possibly a power fit that will fit this data better. So I'm going to quantify this data using power fit and quantify. Now, typically performing this fit takes a few seconds, and I'm going, as I said, that it seems that performing power fit on this data is typically better. It's interesting to know that here that all of these metabolites are being fit in parallel using our scalable cloud architecture. Therefore, increasing the number of metabolites should take the same amount of time. Just to remind you again, this data set is high resolution LCMS unlabeled data, but Quantifit can handle GCMS and LCMS MS data as well. Again, there are approximately 25 metabolites used as standards, but Quantifit can take a lot many more metabolites while still taking the same time. Now that the power fit has happened, you can already see that the fit is a lot better. The R square value has improved significantly, but we still see a warning symbol on guanine, so it might be interesting to actually look and investigate why this fit is bad. Now, this fit, of course, seems bad by looking at the R square value. And what we can do is I can either choose to use a different fit from here and sort of just quantify this particular metabolite. But in this case, I see that one particular sample is acting out as an outlier. So I'm going to reject it here really quickly. And once the sample has been rejected, I'm going to re-quantify this particular metabolite with Quantifit. Now, this will, again, as I said, take a few seconds to quantify this metabolite. And once this has been done, you will see that this outlier has been rejected, and the R square is really good for this particular metabolite. Of course, the warning symbol that was coming beside guanine is no more there. Now, before we talk more about the features on Quantifit, let us talk about all the information we are seeing on the dashboard. At the top, there is the metabolite carousel, which shows you all the metabolites with warnings for those which have bad fits. On hovering the metabolite name, you would also see the fit of the R square values, as I just showed you a few minutes back. Then, of course, there is the curve settings panel, which I just used to change the type of fit for all metabolites. I can do this for a single metabolite as well, as I just did. I can choose to normalize the quantified metabolites by enabling normalization and selecting a metabolite to normalize it with. This normalization is different from ion count normalization. In ion count normalization, the unquantified data for each metabolite was normalized, but here the quantified concentrations are used to normalize other metabolites with respect to the given metabolite. Now let's have a look at the plots. The calibration curve plot is completely interactable. You can choose to zoom in or zoom out, or maybe even reject to look at some data points. Let's just say I don't want to look at the fitted data, just at the standard data and the curve. I can do that as well. Right beneath that is the residual plot, which shows the residual error for the fitted data points. At the top right of this section is the standard data, where you will be able to see the concentration of the particular standards that you have used and the area, amongst other things. Last of all, on the right-hand panel, you see the equation and goodness of fit section, which informs the user about the exact relation between the concentration and the intensity used to fit the data, as well as the quality of the fit, which is represented using three matrices, essentially sum of squared errors, R square, and root mean square error. Now let's move on to a few features QuantFit has to offer. As previously mentioned, you can choose to save your analysis from the top left-hand corner as well as restore an analysis. Users can also export the data before doing using the process tab. And before they do that, they can also choose to scale it by a particular factor. Sean will talk a little more about this in his demo. You can also download the data, as I was talking about exporting the data from here, and put it into any of our other IPython workflows or any of our other apps. We also offer the customizability in terms of allowing you to connect to your own custom IPythons or any of the subsequent apps on Poly using the suggested apps panel, which is at the bottom. Uh, Users can also go to the Poly dashboard and within that the projects and share their project with their collaborators. Now this project that they will share will contain a lot of information about their analysis, such as the IPython notebook, such as the analysis that you did, the parameters you ran this data on, the project files, and the ability to restore all of this data. With this talk, I sort of wrap up my demo and hand over the webinar to Sean to talk more about how QuantFit workflow on Poly has enabled his study.
Great, thanks for the introduction earlier. Today I'm going to talk about how we've used QuantFit to look at the pharmacokinetics of a molecule called cysteamine that was administered to rats. So why should you care about cysteamine? Cysteamine is used to treat a rare inborn error of metabolism caused by mutations in the lysosomal cystine exporter, a gene called cystinosin. As a result of not being able to transport cystine out of the lysosome, it accumulates. It can form crystals, as you can see in the slide, which causes cellular damage in multiple organ systems, but especially in the kidney and the eyes are among the most dominant tissues affected. So cysteamine is a therapy for cystinosis. It's taken for the duration of life, 4 to 6 times a day. And the mechanism of action of cysteamine is that it enters the lysosome and interacts with cystine to release a free cysteine molecule, which is then transported out of the lysosome via cysteine transporter and forms a cysteamine mixed disulfide, which is then exported out of the lysosome via a lysine transporter in the absence of not being able to export cystine itself. So even though cysteamine is useful, it has some side effects. And to date, all existing studies examining cysteamine pharmacokinetics rely on measurements of cysteamine only. So we're interested in understanding the metabolic fate of cysteamine itself and the consequences of the metabolism in different tissues that receive cysteamine when dosed systemically. And to do this, we designed an experiment that contained many rats. Today, I'm going to show you one rat for simplicity. And we dosed this rat with 30 milligrams per kilogram of cysteamine by oral gavage. When we collected plasma at time points from 0 to 360 minutes to look at plasma concentration levels of cysteamine and other metabolites. And all of the tissues were collected at the terminal sacrifice of the animal. We extracted those tissues and metabolomics data was collected using a Q-exactive mass spectrometer in full scan mode. So we could see all the metabolites of cysteamine and try to understand better how cysteamine is being metabolized. So today I'm going to show you how the poly-quantfit workflow has been used to see biodistribution and quantification of cysteamine and related metabolites through a series of the following steps. So I'll show you the raw data processing through Almaven, the normalization of data. So we used internal standards, valine and phenylalanine for injection efficiency, and deuterated cysteamine itself to determine cysteamine identification accurately. And I'll show you the power fit and quantification on QuantFit. Finally, I'll show you the exported data format into an IPython module. So here's the data. You can see here that these are broken down into several different groups. So you can see here's all the plasma samples from 0 to 360 minutes. Here's a number of standards that we ran. So in this experiment, we ran almost 20 to 30 standard curves, and there were numerous rats. So this is just a subset of that data, but it works the same for 20 as it does for 1. And you can see the tissues down here. So you can see kidney, muscle, brain, liver, and stomach. So as you can see, there is some matrix effect with these internal standards. So we added the same amount to each tube, but that also has to be accounted for, and Poly handles that really well. So valine D8, phenylalanine D8, and then you can see cysteamine itself. So after you've picked all of the peaks and quantified everything in Almaven, you can, as Raghav said earlier, you can export to Poly, which is currently already done. And here you can see we are quantifying 5 different metabolites. And so here's glutathione, and you can see the curve fit is good for this one. And then looking at cysteamine itself, you can see most of our samples fall well within range of our standard curve. So from here, after we've done all of the QC and examined all of these curves for our squared values and best fit, and rejected any points with outliers in concentration that might not be on the curve, we can click the process data, and we can see the concentrations of each individual metabolite. And so in this case, all of our plasma samples were diluted 1 to 20, and all of our tissue samples were diluted 1 to 60. So this feature here is nice to really get back to, the value that the final concentration. So it just makes it a lot easier. And doing all of this manually by hand would take many hours. So after this has been done, you can export it to a number of different platforms, and Raghav will tell you about advancements in those export functions after this. But for now, we can visualize this in IPython, and we can see the concentration of cysteamine in a plot of time versus concentration here. So you can see at time 0, the rat isn't dosed, and then you can see the sharp rise of this. And so as I mentioned earlier, the side effects really happen here. So we're interested in what's happening here in plasma to try to better understand how to mitigate some of those side effects. And you can see this sharp decline in relatively low half-life, which makes this a particularly troubling drug to take throughout life. So understanding the side effects better could help come up with solutions that might make patients' lives better. And you can also see the cohort versus concentration in tissue. And so you can see, as I mentioned earlier, the kidney is one of the most affected organs. So you can see a high concentration of cysteamine found in the kidney. So we're further examining the different metabolic changes and trying to understand better how cysteamine itself is metabolized in all of these tissues. And with that, I'll hand it back over to Raghav to tell you about future updates in Poly.
Hi everyone, I'm back and finally to talk about the last section, which is the Poly Quantfit version 0.2. Now, we've been getting a lot of requests from our beta users for a lot of enhancements, and we are actively working towards them. And we plan to release this as the next version of QuantFit. This is what I'm going to show you today are designs that our product team has designed and our product and design team has made over the past few months, and which we plan to ship out in the next few months. Now, and a lot of them were the ability to quantify a lot of metabolites at once, which we are already enabling. We want to. They also ask for easing challenges faced in preparation of the standard metadata. As you saw, that's a slightly complex metadata to prepare for. That is also something that we are working towards, and a beta version of that is already something that is out there in the Kibi lab at Yale. We also, a lot of users ask for customizability without Elucidator support, as well as improved visualizations of the of the quantified data, as well as reproducibility of the results. The last point is something that we already have on Poly in the form of restore and store, a feature that we just introduced a month back. So as you can see, we are already working towards building this particular version. Now, again, to talk very briefly through some key features, the smart metadata interface will essentially pick up patterns between your standard sample and your concentration and save them so that you don't have to build your metadata again and again. So even for different experiments, we will be able to pick up patterns and see that, okay, now this particular sample matches to this concentration, and you will not have to edit and make metadata again and again. Auto quantify for statistically valid best curve fit settings. You will be able to auto quantify your metabolites with the statistically favored curve fit settings. You will also be able to refer to the quantification summary dashboard for a quick look at the results. You will see, excuse me, you will see all the metabolites colored in different colors, and they represent the R square value of the metabolite or of the fit of the metabolite. And you will also be able to filter your summary based on suitable rank metrics like R square, RMSE values, amongst others. You will have the quantification dashboard, a quick view of all your fits. You will be able to glance through all these standard curves using expanded dashboard view. So you don't have to go through every metabolite now. You can just look at this very briefly. You will be able to quantify all your metabolites in one click. This is a feature that is already there on Poly as we speak. This has just come out a month back. And as I said, we are building towards this version already. Peak integration, the ability to visualize and curate peaks on the quantification dashboard. One of the key challenges users face at this point is that you will have to go back to the quantification. You will have to go back to LMaven if you have a doubt about any peak that you have curated. But what if all of that data was already available to you on the quantification dashboard and you could recurate your peak on the quantification dashboard itself? So we are planning to build this functionality as well to enable and ease your life. Smart quantification. We will suggest fits for better fitting, and this we'll do based on statistical measures and in different forms. We'll suggest fits in different forms by the rejecting certain outlier samples, changing weights of your curve fits, or suggesting other curve fits altogether. Concentration plots. You right now saw Sean showing concentration plots on an IPython module which has been hosted on Poly. But what if all of this was present on your dashboard itself? The ability to visualize patterns with respect to cohorts using your concentration plot module. And of course, you will be able to export all of this into your presentation as well. Modules, customize your workflow as you like. At this point, we allow you to connect IPythons, but not actually load IPythons on the dashboard of QuantFit as well. Or for that matter, we don't allow you to, you'll have to go into another app altogether to using the suggested apps panel to and do some different kind of analysis. But what if all of these modules were available to you on the dashboard itself? You'll have an array of prepackaged modules which you can choose from, some of which would also include some very custom exports to Spotfire, Prism, and other drawing tools. Store and restore. This is something that we are already building, something that we are already there on polyquantfit. You will be able to save and go back to a previous analysis. Now with that, I end my session about the next version of polyquantfit, version 0.2, and we are open to taking some questions and answers. Ping us in the chat with your questions or raise your hands. Sean and I are here to answer your questions. Should we? Yeah. Okay. How many molecules can poly take at a time? By Dinah and Mao. Now, polyquantfit can take a lot of metabolites, and there might be slight increase in the processing time, but it has been tested for 100 plus metabolites at present. But that's just for polyquantfit. Other tools like Polyfi can go up to taking 2700 or 1000 metabolites, something that we have tried for as well. Is it possible to use, this is from Fabrizio, is it possible to use it for relative quantization using just the internal standard? Yes, you can use perform relative quantification by selecting your internal standard in the ion count normalization panel as well. And we have another question from Dinah. Why are the standard curve axes inverted from standard analytical chemistry practices? This has been discussed, we came up with this after discussing with some of our users and collaborators. Again, however, if you want this, we can customize this and for you, and we can talk more about in detail for your particular case. Can Poly provide a graphic overview of all the molecules that change through samples, like a heat map? This has been asked by Dinan again. Yes, and there are a lot of ways of doing this. We have the heat map visualization on the First View app. This you can directly go from Lmaven to Poly. We also have pathway level visualization available in a lot of our other tools. Again, you can use this in the Polyfi workflow as well. How to restore analysis from your saved version? This has been asked by Raisa. So Raisa, to restore that, you will have to go back to your projects section. And from your projects, you will go to your analysis. And let me actually show that to you if my screen is still available. Oh, it's hanged. So you can go back to your project section and go to analysis. And upon clicking analysis, the specific analysis that you want to restore, you will get a pop-up about the history of your analysis. There will be a restore button. We can send you a personal text around what exactly this is. So I think that's pretty much it. Are there any more questions that someone would like to know or ask about? We can still answer a few questions. Okay, so we got some unrelated questions as well, so we'd like to answer them. This one's from Ahmad. Just to know, is Lmaven able to make metabolic flux analysis? Ahmad, this is out of the scope of this particular webinar. We are going to do a Polyfi related webinar soon enough, probably in another 2 to 3 weeks. And please do join us and we'll talk more about the Polyfi workflow then. Okay, I think we'll wrap the webinar at this. We just have a few questions that we'd like you to answer. Please answer them at the end of the webinar. Thank you so much for coming today. It was really great to have all of you here. Thank you so much and have a great day.
Thank you.